can probably get going. There's always a couple people that show up
Yeah, okay.
late.
So I will start then. Thank you. Um, I'm going to start on page 192, figure 1, self-tolerance models. But as an introduction, I'm just going to try and summarize some of the things that we've so far learned. And what we've seen is that B cells require T helper cells to be activated. In the absence of T helper cells, antigen can inactivate B cells. We've seen that CD8 cells also require T helper cells to be activated, and that in the absence of CD4 helper cells, antigen can inactivate CD8 cells. So that is really all information relevant to how mature lymphocytes can be activated or inactivated. And then on top of that, we've seen that there is central tolerance as envisaged by Lederberg. And Lederberg had this model that in the lymphoid organs where lymphocytes are generated, the lymphocyte goes through a stage when it's inactivatable, if it interacts with antigen. And if after a time it doesn't become inactivated, it gives rise to a lymphocyte which is activatable. And still, since self-antigens are present before the first lymphocytes are generated, according to Lederberg, you result in a pool of mature lymphocytes which are specific, have receptors specific for foreign but not for self-antigens. So that was Lederberg's model. And are there any questions in the summary that I've just given? So if aren't, we'll go to figure 1. And here is the developmental model at the top of figure 1 on page 192, which is Lederberg's model. In actual fact, people paid rather little attention to Lederberg's model in the middle 1960s on, and that was partly because they found out that very young animals could be made unresponsive to antigen, and they had a model where a lymphocyte could be activated or inactivated, and that meant that there had to be two different ways that antigen interacts with a lymphocyte, one giving rise to its inactivation or paralysis, as it was called at the time, or activation or induction, as it was called at the time, and that is called the environmental model. So there's something in the environment which controls whether you get activation or inactivation. And then people realized that 90% of tolerance was due to the Lederberg type mechanism. So they made a hybrid model, which is depicted there. And what people also realized is that there was Lederberg's model had one major caveat to it, and that was that not all self-antigens are in the thymus at sufficient levels to ablate lymphocyte peptides derived from that antigen, or for the normal antigen. And an example we'll continually use is insulin, because insulin, first of all, isn't present in the thymus at a level where it ablates all the T lymphocytes as they're generated. And also it's very interesting because in autoimmune diabetes, T cells are generated that react with peptides of insulin bound to, of course, MHC molecules, particularly CD4 molecules. So I wondered whether there are any questions about what I've just said. So one thing which is perhaps useful to have up front, just to be aware of it, is something called the priming problem. We'll see that there's some people who believe, myself included, that to activate a T cell, a CD4 cell, you actually require an activated B cell. And to get an activated B cell, you require an activated helper cell. So this gives rise to something called the priming problem. What cell and how does it first get activated? And there are a lot of problems like this in biology. You need ribosomes to make ribosomes. You need ribosomes to make proteins, and proteins are an important constituent of ribosomes. So if you need ribosomes to make ribosomal proteins, where did the first ribosome come from? And that's a very major question that people try to address who are interested in the origin of life. You need life to beget life, so where did life start off? So these priming problems occur quite a bit in biology. It's just worthwhile to realize that what initiates an immune response is not only very important, but it has this conceptual side to it of what starts everything off. And we'll discuss different ideas on that. Now, it turns out that antigen-presenting cells, we've seen, are very important. They take in antigen initially, they process the antigen, and then they present the antigen. And they are actually the first kind of cell to handle foreign antigens. They also, of course, handle self-antigens. They present self-antigens. But it's not surprising that they're very central in considerations of how you initiate immune responses. And in fact, there are 3 different types of antigen-presenting cells. And these all have the same features, that they take antigens in and process them, or internal antigens, and present exogenous and endogenous antigens and present them on, generally speaking, on class 2 and class 1 respectively. But the 3 different types of antigen-presenting cells really have some subtle differences, which actually, when you think about them, are not so subtle and are probably very important. So the first one to be discovered were macrophages. Macrophages are present in secondary lymphoid organs. Macrophages have antigen-presenting cells. Macrophages have co-stimulatory molecules on their surface, and they can take in foreign antigens, and they can present antigens, and they can give co-stimulatory signals. And in fact, macrophages are very important in systemic responses. So if you have an antigen that gets into your body and you want to have ongoing immune responses to it, macrophages are very important. Then under the surface of the skin, for example, there is a whole network of dendritic cells. These dendritic cells have long branch-like structures which cover a very large surface area. The Greek for branch is dendra, and that's how the name came to be dendritic cell. And if you look, there's some beautiful pictures where you look under the surface of the skin, and there's a whole network of dendritic cells with the dendrites covering the area of the skin. So dendritic cells are very important when you get an abrasion, and they have some very interesting properties, and it's really important to be aware of these properties. First of all, immature dendritic cells are very phagocytic, so they take things in. They have a lot of receptors for pathogen-associated molecular structures. For PAMPs, they take a lot of antigen in. They're very phagocytic by various means. They process antigens, and then they can present them, but they have rather little class 2 on their surface for presenting, and they have very few co-stimulatory signal molecules like B7. And under certain conditions, either through a signal or spontaneously, the dendritic cells mature. And when they mature, three things happen. First of all, they become non-phagocytic. They get receptors and they go to the draining lymph node. And they express more co-stimulatory molecules and class II molecules on their surface. And what they really do is they take a frozen picture to the draining lymph node of the antigen environment when they were at the surface of the skin, when they were immature. So, for example, if you get an abrasion in your skin, the immature dendritic cells will take in things like bacteria, which break your skin barrier, process them, mature, go to the draining lymph node, and present in the draining lymph node bacterial peptides derived from bacterial protein antigens. So, and dendritic, that's one example of a dendritic cells. Dendritic cells are in many sites, but what is characteristic of them is that they don't turn over their class II molecules very much. They represent a picture via the peptides they present of the antigens that they experienced when they were immature. So how they differ from macrophages is that macrophages can take in antigen even when they're mature and they express high levels of co-stimulatory molecules. And lastly, there are B cells, resting B cells, when they're not activated. They don't present, they have class II on their surface, quite in substantial amounts, but they express very few co-stimulatory molecules. But once they get activated, particularly by T helper cells, they express a lot of co-stimulatory molecules at a high level. And it's very well known that they present antigen. And in fact, we know that because we had an antigen bridge model of the B cell T cell interaction. And then we realized when then we had to reconsider when we realized that T cells have a receptor that recognize a peptide derived from the normal antigen bound to class II molecules, that the B cell took in an exogenous antigen through its binding to its antigen-specific receptors, process that exogenous antigen, and then presented peptides from the exogenous antigen on its surface so that an activated T helper cell could deliver helpful signals to the B cell. So I just wondered whether anyone wanted to ask any questions on that, on the different nature of the antigen presenting cells.
No, I think it makes sense.
Okay, so we'll go ahead to now one of the biggest questions in immunology. And I'm going to say up front that I feel that what most people believe is very misleading. And I will tell you what I believe. I'm just going to be very straightforward about it. So we're going to go to page 197, figure 3. First of all, in figure 2A, there is a picture of B cell T helper cell interaction, which is going to be useful to us. That's summarizing what I've just said. In figure 2B, we depict a situation where the activation of a CD4 cell requires signal 1 when its TCR binds peptide MHC complex, and also a co-stimulatory signal due to B7 on the APC and CD28 on the T helper cell. So that's a co-stimulatory signal. And that model, as it's presented there, is I call the constitutive model, because it's the APC could be, for example, a macrophage that expresses a co-stimulatory molecule all the time. In preparing for this, I realized I really should have had another picture here. So I'm going to describe it. And as I'm going to describe it, I will explain the background to this idea. And that is that APC do not express co-stimulatory molecules required to activate CD4 cells normally, only if they're activated in some way. And to cut a long story short, and then I'll give you the rationalization for it, Janeway suggested that an APC only gets a co-stimulatory molecule expressed strongly if there's a pathogen-associated molecular pattern from an infectious agent that is recognized by a pattern recognition receptor. Am I still all right here? Yep, sorry, something came up and I just clicked it off. So I'll explain why Janeway came to that suggestion. Polly Matzinger suggested that Janeway wasn't altogether correct, but that actually the immune system senses danger, like heat shock proteins or something, when it's stressed, and that causes the APC to upregulate co-stimulatory molecules on the APC, and that if you don't have upregulated molecules, co-stimulatory molecules on the APC, you just get signal 1 and inactivation. So Janeway proposed this on the basis, I think there's recently, about 3 or 4 years ago, essentially some people gave the Nobel Prize for the idea that innate defense mechanisms control how immune responses are regulated. Janeway initiated this whole idea, but he had died from leukemia a few years previously, so he wasn't part of the prize, but he would surely have been part of it if he'd been alive. And what he pointed out was that if you take purified vertebrate foreign antigens, let's say like BSA in mice, it's actually pretty difficult to get a good response against them. And what you have to do is you have to give that vertebrate foreign antigen in adjuvant, like complete Freund's adjuvant, which has dead mycobacteria in it, and then you get a very good response. And what Janeway said is, we know that for B cells, there are 2 signals involved in B cell activation. We know that for CD8 cells, there are 2 signals for CD8 activation. And we know that 2 signals for T helper cells involved in CD4 T cell activation. And the second signal is a co-stimulatory signal. But what we don't recognize is that when we want to get a response to a purified protein, like BSA in mice, vertebrate purified protein, we use adjuvant. And he called this the immunologist's dirty little secret. It was a very famous little phrase that he coined, and everyone kept repeating it as explaining why he made his theory. So actually, other people have said similar things before, but he really articulated this. And so he suggested that there were things like LPS, a pathogen-associated molecular pattern, and that the innate defense mechanism recognizes these pathogen-associated molecular patterns, which are also on pathogens, but also on non-pathogenic protozoa, bacteria, viruses, and that we have receptors that can recognize these, pathogen recognition receptors, and that interaction between a pathogen recognition receptor and a PAMP is required to upregulate a co-stimulatory molecule on the APC. And therefore, he proposed that the immune system does not discriminate self from non-self, but rather infectious entities from non-infectious entities. Are there any questions on that? Because this is central to, you see, this viewpoint has been prevalent for the last 30 years in immunology. So it's really important that it's clear. So if there are no questions, I'll go on to how Matzinger this was proposed in 1989. Matzinger 5 years later brought attention to some inadequacies of this model. She said, we all know that you can transplant within a species across MHC barriers, and what you get is an enormously rapid rejection due to the major histocompatibility complex being different. And there are no, if you do it sterilely, there's no pathogen-associated molecular patterns. So it can't just be that the immune system distinguishes between infectious entities and non-infectious entities. It must be that the immune system distinguishes more broadly than that. And she thought that when you do a transplantation, it's very traumatic to the tissue. And so she came up with a danger theory. So the danger theory can actually include what Janeway said, the PAMP theory, and so most people, by far the most popular theories are the PAMP and the danger theory. This means that whenever you are going to get an immune response, you need PAMPs or danger to be around, because you need those to activate the T helper cells, and then you need the T helper cells to activate the B cells and the CD8 cells. And all these theories said, and so agreed, that if you don't have any co-stimulation and you just get signal 1 generated, you get an activation of the CD4 cell. So are there any questions on that? Okay, so in 1970, way earlier, a theory had been put forward that actually, when you look at mature lymphocytes, we know that they can be activated or inactivated by antigen. So the question was raised, how does antigen interact differently to result in the activation of a lymphocyte or in its inactivation? And a theory was proposed that if a single lymphocyte interacts with antigen, it's inactivated, and that to get activation of any lymphocyte, you need lymphocyte cooperation. And so this theory gave rise to the two-signal model of lymphocyte activation. If antigen interacts alone with a lymphocyte, it interacts with its receptor, that generates signal 1, and that results in inactivation. If generated alone for sufficient time, to get activation, you need lymphocyte cooperation. And so if you look at 2A on figure 3, figure 2A on figure 3, at the first figure on figure 3, page 197, we've obviously got a situation where you need lymphocyte cooperation for the activation of B cells. And that would be example of this two-signal formulation. And this formulation was actually made before people realized that there was cooperation in the antibody response, for example. So this model actually predicted that the activation of B cells requires T helper cells, the activation of CD8 cells requires helper cells, and the theory also predicted that the activation of helper cells requires helper cell cooperation. So if you look at figure 3, at the last one, we've seen that you require CD4 cells to activate CD8 cells, and the way that this, or the way people believe this happens, is that the CD4 helper cell binds to antigen presented by the antigen presenting cell and activates the antigen presenting cell to then express co-stimulatory molecules. So we've seen actually that this occurs for B cells. We said that B cells, when they're resting, don't have any co-stimulatory molecules, but when they're activated, they do. So that APC there could be a B cell. It could be a dendritic cell, which was immature, and then as it gets activated, because T helper cells can facilitate the maturation of a dendritic cell from an immature state. So that could be a dendritic cell as well. So the model I personally actually believe is shown on page 198, figure 4. And this is called a two-step, two-signal model. And I'll just address a little bit why I believe it. At the conceptual level, I would say actually that I think there's quite a lot of evidence for it. But since it's not very popular framework, it's not very heavily cited. Firstly, one of the problems with the idea that you need lymphocyte cooperation to activate lymphocytes is that in a naive animal, the lymphocytes specific for a particular antigen are going to be very, very, very scarce. And if you say that antigen needs to mediate the cooperation between lymphocytes, you're asking the body to find a way of getting very rare lymphocytes specific for the same antigen to find each other, or antigen to find them and bring them together. So that's called the scarcity problem. And it's clear that if you have mature dendritic cells and antigen, and you have naive CD4 cells, that antigen can cause a proliferation of the naive CD4 cells. So in this step 1, step 2, there's a step 1 step, first step, where presentation by antigen on a mature APC dendritic cell macrophage allows the CD4 cells to multiply. And actually, experimentally, it's known that this occurs, and that these cells, if you've got conditions where you don't get cell cooperation, they go after about 4 or 5 days, they multiply, and then they go into death through apoptosis. They crash and die. So the first step, where you get the cells multiplying, overcomes to a certain extent the scarcity problem. But then to get an activated CD4 cell, according to this proposal, you require a step 1 primed CD4 cell to actually receive other signals from an activated B cell, and that activated B cell has been activated by an activated helper cell to express co-stimulatory molecules, and then you get you get fully activated cytokine-producing CD4 cells. So there are two things I would like to point out about this. One is, where does we have a priming problem in this model? Because we have an activated effector cell activating a naive B cell to express co-stimulatory molecules that is involved in step 2. So where did that effector helper cell come from? So that was a priming problem I talked about previously. The other thing is, I've told you that there are B7 co-stimulatory molecules on APCs and counter-receptors CD28 on T cells. It turns out there are a whole series of different co-stimulatory molecules and counter-receptors which are regulated are very often inducible. In other words, they're not constitutively expressed. And so, and a lot of these different co-stimulatory molecules are expressed on activated B cells, but not on unactivated ones. So there's actually a lot of observations consistent with the idea that B cells can act as APC, and T helper cells can upregulate co-stimulatory molecules on B cells, which are important in CD4 T cell activation. So I'd like to invite questions again. And if there aren't questions, I'll just make 2 further points about this model. No questions? So 2 points. Now I'm speaking as a partisan of this model. So you should put on your skeptic glasses, spectacles. One is that it turns out that certain types of autoimmunity, which are recognized to be due to cell-mediated autoimmunity, are actually treated clinically by depleting B cells. And according to the other models, B cells have no role in a cell-mediated response. So how come B cell depletion is efficacious in treating autoimmunity, like autoimmune diabetes? And of course, in this case, if you get rid of the B cells, you're getting rid of ability to activate T helper cells. So it's not surprising that it is that depleting B cells is a form of immunotherapy. Mind you, it's not a very nice form of therapy, because it really undermines making responses to foreign antigens as well. Secondly of all, there is a mouse model of autoimmune diabetes, which really parallels the human situation remarkably. The NOD mouse, non-obese diabetic mouse, in which autoimmunity arises, and you can study how autoimmunity arises. And what people find out is that very early on in 3-week-old mice, NOD mice, as they become autoimmune, they've got T cells which are specific for particular antigen. And as the mice get older, they find that the T cells, autoimmune T cells, increase in number, but more important in their specificity for different antigens. And this phenomenon is called epitope spreading. So the repertoire, the nature of the specificity of the T cells increases with time. And actually, if you block the immunity generated to the first antigen, you don't get immunity to the other autoimmune targets. So this is really, this phenomenon of epitope spreading is expected on this model. So what this model really says is that lymphocyte cooperation is not only required for the activation of B cells and CD8 cells, but also for CD4 cells, whereas the danger and PAMP models say a single B cell, CD4 cell, a single CD4 cell with the appropriate APC and danger or PAMPs can be activated. So are there any questions on that? So the last thing that, to bring us up into a modern context, is another whole layer of regulation which people recognize is relevant to self-tolerance. So I might just say something as an introduction to this. It appears that nature hasn't made one really good mechanism of self-tolerance. It's like an onion. It's got self-tolerance is an attribute like an onion. It has different layers. We've got the layer of central tolerance. We've got the layer of peripheral tolerance. And it turns out we've probably got at least one more layer, as we now recognize. So this particular story started off with someone trying to do some very analytical work. They took the gene for a protein of the oncogenic virus SV40. It doesn't really, we'll just call it G. So they took the gene for G, and they decided that they would try and make a model for how peripheral tolerance might work. So they made a transgenic mouse, and they expressed G as a transgene, but under the rat insulin promoter or mouse insulin promoter. They're the same, the rat and the mouse insulin promoter. So the idea was that this antigen G would be expressed under the same conditions where insulin was expressed. And we could really look at immune responses to G as a model peripheral antigen. It would be very powerful because we'd have mice which are normal, wild-type mice, and then we'd have the G transgenic mice. And what they found out is that when they made these transgenic mice, in some of the transgenic mice, presumably depending on where the transgene had inserted into the genome, G was expressed very early on in development, and sometimes it was expressed very late on in development. And when it was expressed late, they got autoimmunity to G and to the beta islet cells of the pancreas. And when it was expressed very early, there was no autoimmunity, and the immune system regarded G as self. So that just fitted in beautifully with the historical postulate. All very clear and good. The trouble is, they found out through this that G was expressed partially in the thymus, and insulin is expressed in the thymus. And so actually it was the expression in the thymus that was really important in controlling tolerance. So we're having a model not of peripheral tolerance, but actually in this case of central tolerance. And the people began to realize that peripheral antigens are actually expressed in the thymus, but at too low a level to be really, really reliable. So there was still a peripheral problem. And consistent with this is that the one locus which controls your susceptibility to autoimmune diabetes is a genetic locus that determines the expression of insulin in the thymus. And if it's high, you're better off than if it's low. So there was also an autoimmune disease in humans where people got autoimmune to a lot of different peripheral antigens, essentially. And what people found out in the end was that there's a transcription factor called AIRE, A-I-R-E, autoimmune response element. And this transcription factor controls the expression of a lot of peripheral antigens in the thymus. And if you don't have it, or if you make a mouse which is a knockout for this transcription factor, they become autoimmune very quickly in multiple organs. So what that means is that essentially you've got T cells specific for insulin coming out of the thymus as mature cells at a much higher rate than in a wild-type mouse. And of course, that means that there's much more probability, particularly if you believe lymphocyte cooperation or CD4 cooperation is required to activate CD4 cells. So people realized that there was expression of peripheral antigens in the thymus. So are there any questions on that? So I'm going to talk about one more thing which is related to what I've just talked about, but it's a hell of a lot of information I feel I've covered, or concepts today. But I'm going to come back to something which I mentioned in passing earlier, but so I'm going to summarize the observations, and that is that if you thymectomize a newborn mouse of certain strains at 3 days of age, they come down with autoimmunity. If you thymectomize at 10 days of age, they don't come down with autoimmunity. So people thought there must be cells generated in the thymus between 3 and 10 days which inhibit this autoimmunity. Because if you thymectomize at 3 days, there are enough T cells in the periphery to get the autoimmunity. But if you thymectomize at 10 days, other T cells have come out of the thymus, presumably, which can protect against that autoimmunity. So what they did is they took mice, thymectomized them at 3 days of age, and took thymocytes derived from 10-day-old thymi, and gave them to see whether those 10-day-old thymocytes could inhibit the generation of autoimmunity, and they did. And these T cells were called natural Treg cells, and they had CD25 expressed on their surface, which is unusual for naive cells. In fact, CD25 is a high affinity receptor for IL-2. And when you activate T cells, they respond to IL-2 partly because they express CD25 on their surface. So people thought this is a very interesting kind of cell. It can control autoimmunity. That means, in this case, it doesn't look like autoimmunity is due to the getting rid of the lymphocytes with anti-self activity. It's almost like this is a dominant form of protection against autoimmunity, because we have in our hands these induced Treg cells which can inhibit autoimmunity. So that would be a kind of suppressor or regulatory cell. It's not clear what these regulatory cells are doing altogether. They may have multiple functions, but I am going to say one thing about them, and that is that they tend to inhibit the activation of other CD4 cells like Th1 cells and Th2 cells, which we will talk about next time. They also make two cytokines, IL-10 and TGF-beta. And now these are very interesting because Treg cells are of two kinds. They're the ones generated in the thymus, and they're also ones generated in the periphery. The ones generated in the periphery, people think probably have specificity for gut flora, for example, because they're very heavily present at mucosal sites. And as I said, they make TGF-beta and IL-10. And we've seen to a certain extent that, for example, IL-4 facilitates IgE production and IgG1 production. We haven't talked about that very much. We'll talk about it more next time. But the cytokines that the spectra of cytokines that different T helper cells make determines what their target cells will do. And actually TGF-beta helps the production of IgA, and IL-10 helps the production in humans of IgG4. And these two classes of antibody are first of all produced when you've got chronic stimulation by a mucosal route, such like the gut or the lung. And those two classes of antibody are really remarkable in that they don't activate effector functions, and they actually block the ability of other antibodies to activate effector function if they're there in sufficient amounts by competing for antigen. And IgG4 is a most bizarre molecule and very, very interesting. As I mentioned in the notes, when it's made, it's divalent it's divalent like all IgG antibodies are for two identical antigen molecules. Well, it turns out that IgG4, under physiological conditions, is very labile. It splits into two halves and comes together again. And the result is it's bivalent, but each arm is specific for a different antigen. So it actually becomes a monovalent antibody that blocks the ability of other divalent antibody from interacting with antigen if it's there in sufficient amounts. And very interestingly, people who grow out of allergies, when they're allergic, they make IgE antibody. When they grow out of allergies, they switch the class of immunity to IgA and IgG4 production. And when you successfully desensitize by giving the allergen in progressive amounts, you switch the kind of immunity you get from IgE, which of course is responsible for allergies, to IgA and IgG4. So these Treg cells, or cells like Tregs, they may have multiple functions, but one of them seems to be in deviating the response into benign anti-inflammatory modes. So I think we're probably up on time. I can answer a brief question. That was quite a lot. Please come back with questions next time if you have some. Are there any short questions now? Okay, Brooke?
Okay, thank you.
All right. No questions this time.
No.
It's all right. I know it's really tough, tough times, and I hope that this is really being helpful to people. All right. See you on Friday. Bye.